So it is with great pleasure. It's always one of my favorite things is being able to introduce our own staff, giving talks and tours and things like that, because I'm always so proud of everything that our folks do here at the Arboretum. And so it's with great pleasure that I get to introduce Dr. Andrew Pais, who got his doctorate after starting here. I like to think that it was us who gave him that extra boost that 2 weeks before he actually got his Ph.D. when he started here. That
I don't think my investor would let me go if I didn't have a job.
That's right. That's right. Well, and Andrew worked with one of the Arboretum's biggest supporters outside of the horticulture department here, Jenny Xiang, who is in the plant biology department. Has students who come out here and do work out here. We work with her on research. She's one of those taxonomists that actually understand that the dead things that they put on paper in the herbaria actually started off as living plants, and she passes that on to her students. They actually get to look at living plants, which is pretty great. And so I'm not even going to try and recall off the top of my head what Andrew was talking about. Put phylogenetics at the beginning of any title, and it sounds like you really know what you're talking about. So without further ado, I'll turn it over to Andrew. So.
Thank you. I appreciate the introduction, Mark. Thank you very much. I'm glad some of you are impressed by the title. Phylogenetics is a very smart-sounding word, but it's also a very scary word. Let's put that title up so I can just show you.
Projector turning on. We disconnected the cable for your safety tonight.
So you can tell that I'm a Ph.D. graduate because I'm having a hard time doing a very simple task. There aren't any other PhDs here to make that joke too, right? Okay, so let's start with the title. Application of Genomics to Flowering Dogwood Conservation. Can I turn this down a little?
I will do that in just a moment.
And the biogeography of mock oranges. So the genus, for those who are into the scientific name, is Cornus, Florida. There we go. There we go. Yes. You have a very booming voice. Can you all hear me back there?
Yes.
Okay. So let's start again. Flowering dogwoods, Cornus, Florida. We're interested in the conservation of that genus, that species. And I'm also bundling together while I'm at it another taxa, mock oranges, another ornamental. It's very popular in the Southwest. That's known as Philadelphus. So this is a very ambitious title. I got input to do this, just FYI, from my advisor. So we go all out. Chris told me I won the award of the longest title.
Yeah. With Tony.
Yeah. So if you all are afraid of what that title means, don't worry. I'll try to guide you through each of these words. So I come from a background of evolutionary biology, which you don't know what that is necessarily. We're testing how things are the way they are now. So where does all this plant diversity come from? What are some guiding mechanisms that could help us predict where evolution is going to carry things? So if you're breeding crops and you want to understand, what your offspring might look like, we're using these kinds of concepts. And it could be any species. It doesn't have to be flowering dogwoods or mock oranges. I'm more interested in the questions, but it really does help that I have some pretty looking crops to study. And one person asked me right before this, why did I study these two particular ones? Well, it came down, I had the fortune to inherit the academic turf of my advisor. So my advisor and I study things that are in the Cornelian order, which include hydrangeas, as well as dogwoods are the central genus of that, and mock oranges. So what is evolutionary biology? Let's talk a little bit more about why it's important. You couldn't begin teaching plant systematics or any botanical sciences without having some rudimentary understanding of what evolution is, which is just the change of frequencies of certain genes in a population over time. That could be through that buzzword you hear all the time called natural selection. It could be caused by other just random processes. But more or less what we're trying to do is look at history of evolutionary time and try to predict, why we see the living things that we have now. If we didn't have that framework, we wouldn't be able to explain why the transport systems of gymnosperms are different from angiosperms, while things like clematis have 4 petals versus some of the other things in the buttercup family. It all comes down to just how things were related by a common ancestor in the past. And this common ancestor we try to depict in a picture called a phylogenetic tree, which is that word phylogeny that I had in the title. So it's basically a way of tracing ancestry back to recent ancestors. And with that, I'm going to use those concepts at both the species level, when we're studying mock oranges, and then I'm going to talk about it at the population level with just the flowering dogwood trees that you all know so well. And just to stress this for any graduate student, especially now with tightening budgets, I want to convey to you that these aren't just important questions to me, but these are useful things to pursue from a professional development standpoint. So I'd like to think that I've gathered plenty of computational skills, analytical skills that I think would lend themselves well here at the Arboretum. And I'd like to share some small little projects that I got going on that I think I have an expertise in with biology and with just data science that I think I could contribute to. So let's get started with the first topic, the mock oranges. So mock oranges are prevalent in our garden. We have one here for the raffle. I'm going to introduce to you some we actually collected from my study from the Arboretum itself. So the Arboretum is a very good contributor of research materials for ongoing projects, not just NC State. I'm working with people all the time to send seeds or cuttings of material out to so that they can complete their master's or Ph.D. product. So Philadelphus lewisii, the specific epithet, is named after Lewis and Clark. Think of when they were going out on their western expeditions and describing new species. This one came from that, and you can see its western distribution right over there. If you're looking for it in the garden, come back in the spring or summer. We got it here in bed P06, where the old parking lot used to be. There's another one that we used to have, actually. It's dead according to the records, but I thought I'd still talk about it because it does have a southwest distribution, particularly in Mexico. So we have Philadelphus mexicanus up here. While I'm showing you just these ones that come from the southwest, be aware that there is a global distribution of Philadelphus. So this is looking at the north. If you're looking above the North Pole, this is the Beringia land bridge between Alaska and Russia. And we see a high amount of species in the west. There are some, this is a better zoom in, there are ones like Philadelphus hirsutus, which occur naturally in the east. But if you go across the land bridge, you can find hundreds of other species, not hundreds, I'm exaggerating, dozens of other species in Eastern Asia and some even in the Caucasus region and Western Europe over there. So we have approximately 70 species according to an old description of Philadelphus, and I've gathered at least half of the samples of representative species from what we understand is Philadelphus so far. So how, if we believe the diversity center of Philadelphus is in the southwest of the US, can we explain the history of how things got over on this side of the world and how they became different from each other, eventually diverging into different species? We have a theory. I'm going to talk about one primarily, but one theory that's the simplest is just that they are easily bird dispersed. So think of something being eaten, a fruit by a bird. The bird flies across the ocean and defecates out the seed that becomes, another founder of a new population. So that applies in certain cases. We don't think it's quite what explains the diversity of Philadelphus that we see. Instead, I'm invoking this hypothesis that my advisor continues to study with collaborators from the University of Florida, from Duke University, that's looking at sister species, so species that are very similar but occur in different parts of the world. So I'll give you a couple examples that you might see in your landscape. You have, of course, there's the flowering dogwood that we have. Those have those nice rounded white bracts. But if you look at a closely related species from Japan and China, we also have something known as Cornus, which has more triangular bracts. And I'm showing you some more examples up on the board here. We have Tsuga canadensis. There's an Eastern Asian species called chinensis. We also have sassafras, the one you normally see. There are sassafras in China too. There's liriodendron, or tulip poplars. So there's many things that occur in Eastern North America and then in Eastern Asia. So we try to figure out why do we see those patterns? Why aren't there more bridges between species across the northern hemisphere? Why do they just occur here? So one idea is that there is similar climate, but we also like to think of the things that were happening in history that were causing these things to be where they are now and become separate species. So think of glaciation, so ice ages, pushing plants further south if they occurred across the northern hemisphere. Think of the Rocky Mountains rising, causing desertification out in the west, pushing plant species ranges further east. And now we can see relics of what was a wider range of a common ancestor. So Philadelphus itself might not even be its own intact group. So I talk about this word called monophyly, which just means that all the existing members of Philadelphus now as we know them come from a common ancestor. And most of the time we classified this by visual evidence of what the plant traits were. So we saw that many of these things had carpels or plant flower parts of 4, but there was variability. One unifying trait was that if we actually looked at these under a microscope, we can see that the bundles of DNA are in packages of 13 chromosomes. So this might seem like convincing evidence to say this is different than, say, this other close by genus called Carpentaria, which has 20 chromosomes and is evergreen, whereas these ones have deciduous leaves for the most part. But then again, it begs the question, well, what traits should you really be looking at to describe the evolution and the divergence of these different species? So if I'm just picking one trait because I think it's important and it gives me the result I want, that's not necessarily what could be accurate according to evolutionary history. So we need some sort of unbiased marker, which brings in the new technology we have using DNA markers. And this recent application was, taken for granted at first. We had, 1 or 2 genes sequenced, and we came up with these evolutionary hypotheses just from 1 or 2 genes, but we were still having the same problem. So if you pick this gene, and it's really not telling you that much, but one gene is telling you something other, you're still biasing your analysis. So I've gone one step further, and I've sampled even more genes than the previous researcher here that did a study in 2002. So this is weekly. We published again with Alan Weekly later in my subsequent work. It was at the UNC. So there is evidence that that Philadelphus itself is not monophyletic, and I'm going to show you exactly what I mean by monophyletic. So this is a phylogenetic tree. All phylogenetic trees, we usually like to see things bifurcate. So we want to have one sister species here and another there, and at the node, we expect there to be a common ancestor. And if you go one step further, the complex here should have a common ancestor with the complex here at that node. When you have cases like this, where the DNA evidence is forming a 3-pronged fork, that presents a problem, but that's a hypothesis. So we're asking ourselves, well, how are these branches arranged? Did Carpentaria become a different lineage sooner, or did it become a different lineage within the genus Philadelphus, as represented here by this middle hypothesis, where you see core Philadelphus is on the outside here, and there's a complex of this genus Carpentaria splitting off with these two different species of Philadelphus, known as Philadelphus texensis and Philadelphus murancii. This, again, I'll talk about the results in a sec, but I'll just show you the outline of how we actually generate these trees using DNA evidence. So one thing is just to gather all your leaf samples. So fortunately, I was helping a previous graduate student's project, so I didn't have to go everywhere and collect all these things, but most people don't for the most part anyway. So researchers will reach out to Arboretum, to herbaria, and get samples from all over the world. So the bulk of my leaf collections came from the Arnold Arboretum, J.C. Ralston Arboretum, and Missouri Botanic Gardens. And with those leaves, we stored them in fridges so they don't deteriorate. We basically take out a mortar and pestle and grind up the leaves as much as we can, and then we start adding these buffers in so that the cells don't degrade quickly, and we can scoop up the DNA that's being shredded out of the cells. With that, we get a small little pool of DNA, very, very small. So we have to do something called PCR, where we're photocopying the DNA in many copies so we can actually work with it. And this method that we were using to actually understand what's inside the DNA is called Sanger sequencing, which is one of the earliest forms of getting DNA evidence to analyze. So sequence data here would consist of about 1,000 different base pairs. And we can use a matrix of those thousands of base pairs to compare how similar or dissimilar any given species is from the next. And that's shown by this magic rainbow color here. So that's my creative way of showing you thousands of base pairs at once, where each of these rows is a different species that we collected, and each of these columns here that are different colors is representing one position of DNA on this gene that is common to all these different species. And each of the different colors is representing one of 4 base pairs. So in DNA, there's adenine, thymine, guanine, cytosine. And these are the units of comparison. So whereas you can see most of these things are very similar when you line them up, there are 1 or 2 places where you see a different color for that species versus everything else. And these differences are what we're really looking for to understand and create these phylogenetic trees based on the base pair differences between each of these pairs. And this is one of our good trees that we got. So there's algorithms that can generate this. I won't belabor that point, but we have a variety of hypotheses. So each of these trees is a different hypothesis. And we have statistical ways of deciding, well, which one is the most reasonable hypothesis to print a nice figure out and publish and interpret those results. So I'm going to just guide you through this. These numbers down here, the higher the number is, the more significant and credible we think the result is. All the species, I'm putting the labels up here to show how they're related. Things that are further on the left are diverging sooner than the rest. So we gathered a couple other genera that were also closely related to Philadelphus, just to have a point of reference. So we expect these things to have diverged sooner in evolutionary history than everything else. And like we were testing before with our previous hypothesis, we wanted to see if Carpentaria was diverging first before all the rest of Philadelphus, or like we're showing in this tree, there's a sublineage of Philadelphus murancii and texensis that diverge sooner from the rest of Philadelphus. And the next thing that you see diverging is this other genus Carpentaria. So from a plant taxonomist standpoint, this would be good evidence to advocate for a taxonomic nomenclature revision, which drives, anybody managing records or teaching plant systematics crazy. So I get that all the time now. So every time I print a label out because I think it's this genus, and then a week later I find out somebody's done a study on it and there's a paper out, so the plant list changes, the taxonomic status of something, and they rename the genus, well then I gotta go out, find that record, and advocate for a new label change because it's now called something else. So there are, some things that are more stable than others. I like to think that things have slowed down a bit since we've, thoroughly sequenced the phylogeny of angiosperms, but as we get more and more DNA evidence, we oftentimes do resolve some of these uncertainties. So I'll point out here, the branch lengths of this tree are very small for all these other species. The key I have here, this is basically saying, this says 0.02. So for this length of a tree, it's saying there are at least 2 base pair differences for every 100 base pairs that we're comparing. So these ones have maybe 1 or 2 base pair differences. So using just 5 genes, we can't really tell how these species are related, but we can take a good guess at what we think about Carpentaria in relationship to P. texensis and Nerantia, which we advocate that Carpentaria should no longer be a genus since it's a one species representative. It should just be renamed to a different kind of Philadelphus species. So there's one other thing I will stress before I move on, because I really want to share my dissertation research more than this first year project, but I want to get back to this because this is plant taxonomy. That is what I was trained to do. So most people think of just generating trees and telling you what species is related to what, but I think the exciting part of these kinds of studies are what else you can do. So some creative stuff where you can actually predict what was the common ancestry range of the common ancestors at each node, and also how long ago did these things speciate from each other. So this is a different tree where it's actually dated to time. So we start with 0 millions of years ago, and we go, the further back on the left you go, the greater amount of evolutionary time has expired. So this is 50 million years ago. So we're actually able to utilize what we understand about mutation rates in DNA and where we expect, how many numbers of bases to have changed over millions of years in combination with fossil evidence that's out there. So we can reference this back to what we originally asked ourselves. Does Philadelphus originate from the American Southwest? And how long ago did it diverge from everything else? So I have a tree here where, again, you have your outgroups of other genera outside here, and then our common ancestor of Philadelphus, Carpentaria, and the rest of Philadelphus occurs right here. So this would be at 30 million years ago, around the Oligocene period. So this was a little bit warmer, and as you get to 10 million years later, it starts to get colder where you have some of those glaciation events that are isolating different populations which will later become different species. So it makes sense that when you go past 30 million years ago, there's a wide radiation of different species resulting after they're becoming isolated. Another neat fact to point out is P. merantia and P. texensis are primarily from Mexico and the Southwest. So this blue color here is showing a combination of C and D, which is Western North America and Mexico. These species here come from a variety of places, Asia, Eastern North America, everywhere. But if you keep going further back, we can find evidence of the range being much more limited to the common ancestor of these Western species and everything else. So this pie chart is representing the probability that the common ancestor was belonging to a certain distribution. So the one with the highest slice in the pie is C, which represents Western North America. And then these other slices of pie still include Western North America and Central America. Whereas, there is some small evidence for a distribution in Eastern Asia. But we have credible evidence to say, likewise, that this is most likely the origin of Philadelphus itself. So I will conclude. Basically, we revised and completely dismantled the previous classifications of Philadelphus that were based on just plant trait evidence that you see by visual inspection. We advocate that there should be a new way of classifying Philadelphus where the genus Carpentaria is no longer a genus, becomes part of Philadelphus. And then you have this one unique lineage of Philadelphus that is just those 2 Western species. And then there's everything else that we call core Philadelphus. We believe that these 3 lineages likely evolved and speciated in the American Southwest, And as it got colder and things became isolated, you saw more radiation. And as lineages traveled across the land bridge, there was more room to diversify, and that's why you see most of the diversity of Philadelphus that we do today. So there's a lot going on there. So I should have thought, well, maybe this is too much for me. Maybe I need to, cut it up here and go with a master's and not go with a PhD dissertation. But this was just the side project. So I was, getting my credentials. I was getting my publication with, his name is Yulong Gao. So he's working at RTI now. So he got a master's and he went to a bioinformatics program at NC State and later graduated. So we're, both glad to have been under the tutelage of Jenny Xiang and Alan Weekly to collaborate in this. But I had to eventually, strike it on my own and be the primary author, which is now the second part of my talk, focusing on the flowering dogwood tree, which is our state flower in North Carolina, of course. It's the state tree in Virginia, and as you hopefully know, it's a southern icon. So unfortunately, the flowering dogwood tree is under threat. It's very popular in an ornamental setting, but the cost of maintaining it in a nursery has increased more than tenfold. So you have to have fungicide treatments, you have to breed better cultivars that you can market. The landscape is very different for nurserymen planting dogwoods than it has been in the past. What other people don't really think about is the importance to the ecology for flowering dogwoods that just occur everywhere around the US. So these species are well known for contributing calcium to the soil. So they degrade very quickly when they die. Their leaves fall off very easily. The fruit that they get is picked off by birds. They're browsing feed for deer, invertebrates. Microbacteria will also eat the leaves that decompose in the ground quickly, whereas other things, like the second closest contributor of calcium, are white oaks. They, for whatever reason, have their calcium washed out sooner. So if you have any leaching going on where water is moving nutrients down, the calcium that's donated by oaks is less likely to stay. So calcium is important for most organisms, so like bird shells, insects, exoskeletons. It's very essential for cell signaling. So it's one of the cruxes of natural ecosystems, and without that, we'd have a very different landscape. And unfortunately, we are seeing that functional contribution of dogwoods disappear as this flowering dogwood anthracnose disease is becoming more prevalent. So those of you who might have followed this in the past will understand that this disease was introduced in the 1980s. It's believed to have spread from a close Asian relative, and since it's been introduced, it's been working its way southwest through the Appalachian Mountains and extending its havoc. And whether or not it's going to continue is one question. So nurserymen are crossing and breeding for more resistant dogwood anthracnose cultivars, but there's also other things that are now becoming more dominant, and people are struggling to find things that are both resistant to that, and on top of that, scooping up a tree that likes to occur in the shade, and then being very sad when you put it out in an open canopy. So I was just talking to somebody who was saying, dogwoods are very persnickety things. They're nice for like a few days, but then a lot of them can start looking like that if you're not taking good care of them. So there are a lot of challenges, and what my dissertation was about was finding the treasures in the wild landscape that could be utilized by other plant breeders that we're collaborating with. So we have collaborators from Rutgers University, University of Virginia, University of Georgia, and University of Tennessee. I got the fun part, which was going out and walking out in the woods. So this is the range in dark of the flowering dogwood tree. This is the red counties, our U.S. forestry reports of where the disease has occurred, believing, at least for the eastern range, to have been the epicenter of introduction of this exotic disease. And again, we are still tracking and monitoring how far this disease is spreading. It's an ongoing issue. We were worried that this tree was going to go the way of the chestnut. Fortunately, the Pais of progress for this disease has been much slower. So real question is, what is holding back the disease? Is it just environment, or are there actually specific genes in populations in the Southwest, or even those that are persisting in the mountains, to say that, well, maybe this tree is living because it has some actual inherent resistance to anthracnose disease? We could also ask, if we're not just looking at disease, what previous environmental factors have contributed to the evolution of these drought tolerance, temperature tolerance, or disease resistance genes? And are there any associations that we can pull out? So is there a correlation between one version of a gene and a specific environmental gradient that you see across the landscape? I will show you a visual way of looking at this. So this is an example. Let's think of a gradient of temperature. So on the right here, you see we have a representative of a colder climate, whereas on the left here of this diagram, you would have warmer temperatures. And in a normal temperature setting, you might see just a random mixture of red and green genes. So I would say that the red genes in this picture are representative of something that might be adaptive to higher temperatures. So in normal temperature, it's not a problem. As things get warmer, you see that there's more dominance of this heat-tolerant protein, which is one of the ways we identify evidence of a particular gene being important. The same concept can be extended to dogwood anthracnose disease. So on the right here, we can say that there's very low occurrence or prevalence of dogwood anthracnose, whereas you go further left on the gradient, we say there's higher prevalence of disease in this area. So the red genes in this case, we believe would be resistance genes encoding for proteins that we think respond to pest or disease to identify and reduce them or kill the pathogen. So that red gene becomes higher with this disease gradient increasing. There are other ways we statistically test this. So we look at distributions of what are called FST indices. So the higher the index, the FST index is, the more dominant one version of a gene is versus another. So we're looking for those outliers that have really high values, whereas everything else that's not undergoing natural selection or evolution is high on that distribution. We incorporate other models which can control for the environment's effect on how bad a disease is, while actually testing the association of a gene on something like disease resistant. And we also control for the population structure. So the more farther away you are, we'd expect the more different you would be from another tree. So trees on the west side of North America, we expect would just be more different than the ones on the east because they're more isolated, so they're sharing less genes. So we have to have a way to control for that in our models. And this is a lot of a big approach. So we started small. We just looked in North Carolina, and that's what I'm going to mainly talk about, is the pilot study I did during the first 2 to 3 years of my dissertation. So we picked this area because the environments are very different, as you know, in North Carolina. We have mountain populations, which have shorter growing seasons, higher elevations. Piedmont areas, which are typically drier, and then the coastal region, which has different soil types. Another reason why I started with this pilot study is, as I can tell you right now, all the stuff I'm already talking about, studying all this at once was a little bit overwhelming. So this is the range that I have sampled at the end of my dissertation, even getting samples that are a disjunct population in the peninsula of Mexico. The pilot study itself, again, was limited to North Carolina, but you can still see at the national scale where the differences are in environment. So disease, for one, is greater in the mountains. You can see here using GIS, there is differences in growing period between the coast and further interior, of course rainfall, and again soil type. I will say this was a lot of work, and it's very, I don't want to sell myself short, so I'm going to just tell you all the stuff I did real quickly. I was collecting all the environmental variables. I was collecting all the trait data of the plants. I was looking at experiments to test, well, how resistant are things to drought by looking at salt concentration in leaves. And then again, I'm taking this small task of just sequencing, 1,000 genes. So the previous study I did, I sequenced 5 genes. And now we're getting at what I call the genome level, which is almost all your base pairs. I'm not getting all that, but I'm getting good representatives. So we're talking about thousands, and if you don't filter, millions. So someone put it, I was a one-man army doing a giant dissertation project that could take 8 years. And as I can see, I'm already approaching the time allotted I had for just this segment of the talk. So it's a very big project. I won't stress that enough. So there are all these different environmental measures I got throughout the site, the sites I sampled, as well as using GIS data as I've shown you up here. And there's other things such as measuring chemicals in the leaves and measuring plant health. So I was trained with forestry health monitors to first visually inspect trees to see what percentage of their canopy was diseased, where scores of 1 are basically they're dead, there's a lot of branch dieback and blotting and necrosis, and where trees that have a score of 5 are pretty happy. And then we had other ways of looking at the quantity of the microbes in the cells using some genetic measures, but I won't stress that. This is the most intuitive way that we initially started with. The sequencing method, again, I won't stress too much about the details, but we're at the point now where we were getting a draft genome of the dogwood at the time, but that's a very expensive endeavor, so we were using a method that just takes little snippets around the genome. So you get little islands of comparison that you can look across trees. So you can line them up and say, this sample has this version of a gene and that sample has a different version. That's the basis of what you're comparing. So you're looking at different versions of a gene and you're looking for ones where they're correlated, certain gene version is correlated with a certain environmental effect. So with the North Carolina study, we started small and then we expanded to many more hundreds of samples for the national study. And I'll talk more about that result here. But we're showing you, at least with this example, rather than finding just genes that are under natural selection, we can also characterize, how genetically different they are across America. If you were to pick a dogwood in the southeast versus the southwest, what genetic differences more or less could you see? So it's kind of like doing something with Ancestry.com or 23andMe. You want to predict where you came from based on the genes you have. So we can see clear patterns, and there's many different models I did, but we can see there is a nice overlap of the coastal plains with one genetic cluster when we analyze the base pair differences of all our tree sample. We can also see there's a very nice overlap of this blue region, which corresponds to the hot continental mountainous region. And coincidentally, we think it's very interesting, it's also overlapping with that disease range that I was talking about, where this is the most disease-ridden area for dogwood and anthracnose. And of course, we expect that one disjunct population to be very different. It's very far away. And if you look at it in our garden in the spring, you can see morphologically it's got pearly bracts. So that wasn't a surprise. But these are some of the similar things I was talking about with talking about how species are different in Philadelphus, but now we're doing it in the population level for flowering dogwoods. And really what we're getting down to is looking for particular associations of individual genes to environmental gradients, suggesting that they might actually be under selection. So we found many genes were correlated to these factors of temperature, so minimum temperature in January, the growth period average, mean temperature across 50 years, and the frost-free period, which is estimated from some of those variables. So temperature is very clearly a major factor in shaping the evolution of plants. And we can see that if we're looking at the frequency of alleles, or gene versions, changing across these gradients, which is the x-axis. So I'm just pointing out 3 examples of a way we have of graphing this, where, say, potassium, higher potassium levels, the genes that we believe are under selection are actually responding to that gradient, whereas other genes that are not believed to be under selection are staying relatively flat. Same thing applies to precipitation in July and frost-free period. There is a marked difference between the genes we believe are under selection and the genes that aren't. So I'm going to just share real quickly one candidate we have that we're going to explore further. So there are other prospective graduate students, if you want me to recommend them to an advisor that can take up the mantle of this project based on good targets that I think are worth exploring. One of which is a lectin receptor kinase gene, which is encoding for a protein that occurs on the cell membranes of plant cells. So diseases can work their way through the surface here, and plants need to have a good way of recognizing them so that they can either trigger pathways that will create proteins that will kill the disease, or they can create byproducts like chemicals or metabolites, which can also deter pests. Think of like the mustard seed taste, or tobacco nicotine, that can deter infection. We believe through all the evidence we've gathered that this is showing notable patterns of being under selection based on these correlations to disease that I was talking about. More interestingly, I mentioned the second part of my dissertation, I was actually extracting these chemical compounds using what's called chromatography mass spec, which produces these nice little curves, which, like the sequence alignments I was showing you early on, you align these, there are many different colors responding to different individuals, you align them by their properties, and you see what are the differences in the abundance of the chemicals based on the size of those curves. So the more area there is underneath the curve, the more concentrated that chemical is in a given tree. So we were correlating this to our candidate gene, that lectin receptor kinase that we believe is important, while controlling for environment and monitoring how this level of that chemical compound is related to disease susceptibility. So this chemical compound is belonging to a class known as iridoid glucosides, which are well known to be deterrents to disease infection. In fact, there's one compound here called cornolactone that was actually structurally investigated by an independent researcher for flowering dogwood trees. So we think this is a very valuable candidate that warrants further inspection. So the evidence by itself might not be as credible, but all the evidence coming from my first chapter where we just studied North Carolina, plus the regional study where we still found disease associations to this gene in plant health, and the second chapter where we actually found associations between that compound and that gene in plant health. They all tell a very compelling story that warrant this to be one of the subjects for further study. So when you're dealing with thousands and millions of genes to investigate, it's important to have studies like this where you can narrow the target so that other people can actually do some more experiments on this and even do crosses between trees where you can look at a certain gene to see if one cross has a higher level of this compound than another. So it's a different way of looking at plant health. So seeing the interesting results in the big data set is something that I think I can do well. And I hope that I can find those same interesting results in the big data sets that we have here at the Arboretum. So while I might be working with a very different data set, I think data can always tell a compelling story if you work hard enough to filter it and try to find the questions that you didn't even know were questions. So there are a variety of data sets. It's easy to get caught up in our records. And I'm going to do a couple of demos to show you what our database looks like for anyone who is interested. And if any of you are interested in data entry, I know it's a special kind of person who wants to volunteer at the garden and sit at a computer, but that's always something I'm happy to facilitate and give you some projects to do. Our records are robust. That's one of the strong points of this Arboretum is how well we keep records. We currently have this many living things, just planted in the garden that have records in our database, 7,000 and a half of which are unique taxa, so meaning there could be unique cultivars, unique species. And if you're looking at just even the dead records, we have an abundant amount of things that have died since 1976, which, maybe I shouldn't be advertising, but I think it's, maybe that we still have that. We can revisit that. And I am actually interested in visiting those dead records. All these data records have these attributes, and I'm showing you in this graph all the different tables we have linked up to our collections. And this collections table itself has up to 100 different columns in it. So there are plenty of stories you can tell sitting right there. You don't have to do more experiments or spend more money when it's sitting right in front of you. So I'm going to show you one example of something that's just right there I noticed when I was playing around with the database. I was looking at our database records, so this is one screen of one of the layouts we have to look at, is it mapped in our system? What are its coordinates in the garden? We have some scripted buttons where we can report it being as a dead plant through further review. We can tell you if it's been moved somewhere else, if we've propagated this plant, or we sown seeds from it. So there are a lot of different things that I manage on a day-to-day that just help report records and keep them as updated as possible. But then there's this other stuff. When you click the dead tree button, it automatically fills in the date that you noticed it being dead right here. And when you click the shovel button, you're taking something that was reported out of the nursery and into the garden. It fills in very conveniently the last date it was planted. So I was like, oh, this is a really cool data set I could use for creating something that ecologists love to do in intro to ecology classes. So I taught ecology. This is something we did with students. We'd go out to the cemetery, and being real morbid, we would tally up whether or not people we were looking at were male or female, and how old they were when they died. So that's just a way that insurance agents, for instance, can try to predict when they're going to charge you the most insurance, is when are you more likely to die. And I think that's a very interesting thing to look at from just a maintenance standpoint. If you're constantly losing a certain taxa, maybe you want to know where is it most vulnerable in its life cycle. And different species behave different ways. Some species have high seed mortality, where the percent survivorship of things drops dramatically. So the y-axis, at the initial point of a study, if you're doing this, you'd expect everything to be alive. And after time expires, say for this hypothetical species in 7 years, everything should be dead, right? So I'm going to use those different measures of birth versus death. In this case, when it's planted and when it's dead. So I'm assuming that when things are planted, that's a good unit of observation. They've been sitting there an equal amount of time in the nursery, which I understand is a simplification. But still, we can get some rudimentary picture of how things are surviving in our garden. Because things are dying all the time. Wouldn't it be nice if we could improve how we care for them? And I just thought, the t-shirts that we sell right now, we have a good example of Elysium. If you look on the back of the t-shirt, I was like, well, let me just find all the ones that have died. Specifically, I just wanted to play with the dead records. I wasn't even interested in the live ones. And I'll say, okay, let's tease apart, for about 20 of these records that I have reliable evidence of knowing when they were born and when they died. What is the pattern? So what does that survivorship curve look like if I isolate that in my found set and I create some scripts using connections outside of the database to just click a button and then create one of those curves. So the end goal is to have other people on our staff be able to just click a button and do this. But I just want to test it out for myself. So I have a link here. Let me just get the script started. We'll do a couple demos, and if they don't work, then we'll move on. I also want to just impress you with my fancy terminal skills. I have the commands already set up. So what I'm doing now is I'm just connecting to this external database I have out there in the cloud that I've gotten to hook up to our database. So this is using Python scripts. And it's pulled up my little cloud share space. So I'm going to go back to the link I have set up, where it's actually going to command our database to move that information I have on the screen cap and take it to my script center where I'm going to perform the script. Oh,
You have to go to presentation mode, I think, to click on the link.
oh yeah. Good idea. Right. See if I can navigate my mouse over there. All right, so it's pulled up script space. Without looking at all these other folders, we'll go to my script place. We're going to go to the Python folder that has it, and I'm going to click on it, and it's opening up, what the script looks like.
You only have that on your computer, Andrew. It's not mirroring.
Yeah, we can put it on the server if you like.
Well, you can just push it over to the side so we can see it on the screen up here.
Oh, sorry. Let me get out of presentation mode. That's right. Okay, there we go. See that? It's in the bottom. So I'm going to click this run button. So it's running the script now. You can see it thinking about it's moving stuff out of the database. And now it's implementing the script. And now it's generated that cool curve. So I have about 20 records of Elysium. I'm just assuming, there aren't too many differences between Floridanium and other hybridized cultivars. But we can see something, it's comparable to one of these life history curves. We can see a high drop-off of mortality, and the rate of death is pretty constant. And then those that make it past 3 years are made in the shade. They're happy. They're going to live until 7 years. So there is a bias here since we are starting with records that are all from 1976. There are other scripts where we can try to predict how long things will live. It becomes a little bit more complicated. But I just wanted to show you the concept of taking records out and then moving them where you can click a button and start a script. So next thing will be to just embed this on our server so you can do it right from the server and not have to worry about taking it somewhere else. So this looks pretty bleak, but that's actually the way a lot of plants are. You don't have to worry about how well we're taking care of our plants. When you look at these kinds of curves, plants typically follow what is known as the type 3 curve, the one that looks like a water slide going straight down at first and then leveling off. Whereas humans are the ones where we would hope that infant mortality is pretty low. So a lot of things still survive at a high rate early on and then just reach their natural life expectancy. Whereas our results of Elysium in this case are showing something very interesting, that it's a pretty constant rate from 0 to 3 years, and then it kind of slows off like the regular survivorship curve we expect with plants. But if you look at things like oaks that shed tons of pollen, have giant mass sessions where all these acorns get thrown on the ground, they get stepped on, they get scooped up by squirrels. There's a very high probability that that acorn isn't going to make it and carry on its genes to the next generation. So very different species have these different life curves. So other than that, let me move on and show you some less ecology-specific things and more just informative things to look at if you want to understand more things about the plants you study or that you're working with. So I'm going to use other outside databases, hook them up again to our database system here, and try to understand some basic questions, such as for this example that I'm showing you now, is the current name of a given Philadelphus species accurate? How can we easily click that just to test that? Because that's half of my day, is getting samples donated or planted and checking Is the nurseryman calling this the right thing, at least at the genus species level? Where does it fit on the tree of life? Right now I've only hooked up one small portal, which it doesn't tell you at the species level, but it's a nice, overall map of how things are related in our records. And then one thing I thought was interesting is we have all these volunteers who try to follow back stuff we get from index seminar collection trips and fill in the collection details. But wouldn't it be nice if we could just have an easy way to click a button and go to records based on the name of the record to see what other people have reported about the distribution itself, so that we aren't typing all these different things in. So I will start with the valid name one, which is very easy using FileMaker. Escape. So this is actually me opening up the database system itself like I would on a day-to-day basis. I log in, I go to plants, or I can go to the names. So if I'm investigating just the name of something, let's say we pick one of the things I talked about, Philadelphus lewisii. It's been some time since I talked about that species. I haven't actually gone back and asked myself, is that still a correct species name? So I can just click the button. It gives me all the examples we have of that species in our names record. I can look at, how's Philadelphus related to everything by just clicking this link that takes me to a different database portal, and it shows me where Philadelphus is in relation to the rest of the tree of life. You can move around just like you would if you were on Google Maps. You can zoom in and out. You can keep going until you get to eukaryotes. So yeah, this is just one quick example. And getting back at the question, is this a correct name? I have this hooked up to an API with the plant list, which is a credible resource we often use. And it searches right away. I don't have to open up another browser and type the name. It just pulls up the genus species. And in this case, it's showing me the different records I have for Philadelphus lewisii. And here I can see the very top result. It's an accepted name. So I can move on, and I can even just click next sample I have if I want to test that one. These are all Philadelphus lewisii, so they'll be the same thing. One thing I will show you, the distribution map. You don't have to just do it to one species. So if I want to look at the range of all Philadelphus species at once, I can, for instance, just click on the collection list. I can do some quick search. So I'm going to pull up all the Philadelphus we have living that also have a species name. So. Right, I'll put them in list mode so you can see that. I'm going to omit all the gone records at least and alphabetize this. So I'm going to also just quickly, so I have less things to search, I'm going to remove some duplicate records. It just goes quicker if I do that. So what we're going to do is we're going to generate a map of all these species just to see what is the range of Philadelphus across the world. So we have about 10 things in our record, at least in our database. So with that, I'm going to start a quick script that we have in a button here. So I can just, oh, that's the name file, sorry. I have to do it from this window. All right, I'm going to click this button right here, which takes us to our source tab. So oftentimes if Mark's going on a collection trip, he'll fill in collection number here, the province it's from, source details, coordinates if we have them. But if you want to just get a quick map, I thought it would be nice just to click a button. And if this is hooked up on the server, it would work full through, but it's thinking about it. So I have a crappy internet connection, so it'll take a while to see it spinning here. So it's moving the data out of just our database, and it's moving it to that portal I have in the cloud where I can access it and implement my own scripts. So my script will take that data that now you can see gets exported, and I can send it to another database known as GBIF, which has a lot of open source information about any species you want to look up. So it can pull up distribution ranges, it can pull up predicted ranges where they'll be in the future. A lot of neat things you can access if you can just get the data out there. So I script to that, I'm implementing it, it's running, hopefully it'll work. Sometimes the website goes down and it doesn't work. That's one thing about tying yourself to other databases. You know, bad weather can ruin your project for a day. So it's thinking about it. There we go. And I'm going to open up the results. So let me pull that up there so you can see. We just generated a distribution map of where all these Philadelphus species occur in our record. Now, I'll note that I haven't implemented the filters that pick the highly credible results that are coming out of herbaria, our expert taxonomists that rate these things. So sometimes it could be somebody with a photo saying, oh, I think it's that. So you got to filter out that. So I've pulled up things where I found Cornus, Florida, naturally occurring in Europe where it doesn't. But the idea is, you can at least pull up these figures on the fly if you want to just talk about one taxa group, maybe for another presentation in the future. So it's a very convenient way of just getting all that information without having to do a lot of searches. So with that, I'm going to conclude my talk soon, I promise. So many exciting things to do. Now Mark's asking, what do you do at your office? I promise you these are weekend projects. The real cool thing I do want to talk about is talking about the validity of the plant data or the plant identity itself. So again, as you can hear from this talk, my background is using molecular data to understand how things are related. So why not use those same techniques and resources to understand if some of the things in our records can be pinpointed down to species. So currently, as it stands in our records, we have 103 living things with an affinity status, meaning that we think this collection is similar to a certain species based on its morphology when it was collected. So we've collected, say, seeds or cuttings from an area in China that's very difficult to get back to and verify the parent plant. Similarly, we also have many species where we don't take a guess at what the species is, we just assign it to the genus. And then there are just those records that are sitting in the greenhouse where like, I don't know what that is. So these things can be identified. I am a plant taxonomist that is classically trained to identify things. But one of the most informative things you need to identify things to species is flower and fruit. And as you all know, we're an arboretum, so we have many woody trees that have very long lifespans that won't get fruit and flowers when you want them. So how can we get to the answer quicker? One solution I have to this is what's called DNA barcoding, which is using some of the same genes that I was studying in Philadelphus, where you only need about 100 to 600 base pairs to actually get you close to the right answer. So this barcode, if you get the DNA out, think of it as scanning it, searching it against a database where people have been meticulously getting DNA for all the species they can in the world and seeing if there's a close match. This is done and famously with conservation biology. So a lot of people are worried about what they eat in their food, if it's really what it is, or if it's an endangered fish that was caught, in large bycatches. So unfortunately for seafood sellers, we're learning now that in some areas, half of the time what you're eating is not really what you think it is, as evidenced by just this recent study. So that's what we're doing exactly with plants. I have a good laundry list of things I'd like to sequence on behalf of the Arboretum with collaboration from my advisor. The first task I've been doing right now is just sorting out what actually has information on the database. So I hooked up a sequence database owned by the national government's funding to tell you whether or not, say, this unanimous species It doesn't have a ribosomal gene that's very informative, but it does have this chloroplast gene, which is very diagnostic to what the species identity is. So using that list of 100 things and help from one of my very dedicated volunteers, we now have a bundle of records that we can recommend for my advisors class, who they study plant systematics. So one of their class projects is to gather plants every year and try to identify what is that plant. So in the past, we've studied stuff like calycanthus out of the Arboretum, or we've studied things that just the student was interested. So they just go in their backyard and they find out that this plant that they didn't know was a camellia is a camellia. So we think we can use that inquisitive nature to solve some actual questions that we have and apply that to day-to-day things at the Arboretum. So they would extract the DNA, they would photocopy it to have quantities to work with, so all those reagents are covered by the class. The reagents also, they have resources where we every year are working with the Genome Science Lab at North Carolina State University, where they can sequence and get the identity of a sample at about $10 per sample, just by sequencing 1 or 2 genes. And I am trained to lead these workshops. I'd be happy to do it again, even on a volunteer staff capacity, to show people how to use this free DNA subway tool, where the sequence center emails you the sequence files, you get the data, you upload it, and then you generate those neat phylogenetic trees I was showing you, and you can predict what the species is. So if we have plants, we can figure out what they are in our Arboretum. If that sequence isn't in a database, well, we're contributing novel data to this database owned by the NCBI GenBank, and also contributing to the scientific community where they might be wanting to get sequences of other samples for their study. So that's a very big project, ambitious project. Where I'd like to scale up to that. I'd like to find more collaborators, maybe Duke University, UNC, anybody in the Triangle who's interested in pulling in on this, looking maybe for donation resources. But I understand this is just a pilot demo with my advisor using her resources and class resources. What I'd like to do, just at the larger aim of all the things I'm doing here, is to make our database more accessible to our research partners and also to you, the public. So I'm going to cap off with some really cool things that you can just take away on your way out of the plant buggy. One thing just to keep in mind for, I'll be talking about this down the road when we have it set up. We are implementing a new web portal to access our records. So right now you're going through our web page and searching for records and pulling up a map of where it's located. We're going to have an interactive map where you can zoom in and zoom out of all our records and search for particular ones or look at all the plants in a particular bed. That's a project that I really give more credit to a GIS student we have at NC State University who I've been collaborating with. And that's an upcoming project to get data out to you. Something immediately I can do to get you out the door, other than the raffle plants, is putting some nice little labels on the plant buggy outside. So you might have noticed last time you were running by a parking lot just to get something off the cart, we have now these little QR codes in the middle of our plant labels on the buggy. So I've created a script which can use a Google API and pull out the species genus name from our website and then create a custom QR code for it and slap it right in the middle. So if we want to print 30 labels at one time, you click this button, it's going to generate 30 labels where each of the species, unique species, is going to have a unique barcode taking you to our website so you can learn more about that plant. We don't have to squish as much information on how to take care of it on the label itself. So give it a try, take a shop around, don't be shy, just buy.
I appreciate that. It's nice to have that tag on.
Yeah, so thank you. These are I'm cutting way over time, so I'll leave this up here just to acknowledge my vast amount of collaborators in all my dissertation projects here at the Arboretum. So thank you all for your patience and listening. Have a good night. Yeah.
So a question, so what is usually the life cycle in years of a dogwood?
Oh yeah. So I've measured them more, not really thinking about size, but I've been measuring the diameter by height.
Age.
Age, you know, I'd say you'd see ones that last 20 to 30 years, but initially there's, you know, high die-off. So you see all those little shrubs of dogwoods when you walk in the woods. Most of those don't make it. They're trying to, you know, pull out above the understory, and if they get crowded out, they die very easily. But I've measured ones that are, you know, 30 inches in diameter out in the coast that are they're like just on the edge of the forest. So they're growing, again, plenty of sunlight, but they're not getting too much sunlight. And that's where I noticed the most success in dogwoods is when they're on cliff edges, or they're near streams or trails. They tend to do very well and live longer there than they would in the middle of the forest canopy.
I have one at my house that's 40-plus years old, and I wondered
Oh, yeah.
how unique that was.
Yeah, well, in a human landscape, you also get to pay more careful attention to it. And that's one problem that forestry managers have, is that, you know, dogwoods that are cultivated and maintained in our yards, you know, we can identify disease right away and apply fungicides. But what is the natural resistance of trees that really we don't have resources to take care of? You got any questions? Like,
Take a look at the national champion dogwood about an hour east of here, this little over 4-foot diameter.
oh, that's cool. Oh, no, I am not.
It's pretty special.
Yeah, at the time of my study, I was trying to like stay as far away from cultivated things as I could, and you could pick it out on that graph I had of the national distribution, and their colors according to their cluster. I actually omitted some dogs because I was able to confirm that those had close affinity to things like Appalachian spring, things that are commonly planted now across the United States, and you can pull that out through genetic data, which is, you know, promising.
So going back to your Philadelphus, since Carpenterias are so different looking than the Philadelphus, why not take the Mearnsii and the Texensis and make those a different genera?
Yeah, and that's getting to the whole lumper and splitter argument. I guess our reasoning is that, you know, it's one change versus two changes, simplicity. Now, that hasn't been formally done. I was merely presenting the results and making the recommendation. Somebody honestly, you know, could go with that and do that. No one's done it yet because I will acknowledge there is still some statistical uncertainty with our other trees. Like we have trees where Carpenteria isn't diverging later than Mierentii texensis, but instead the common ancestor of Mierentii texensis and Carpenteria is diverging from the rest of Philadelphus, if you understand what I mean. It's just how the branches are arranged. So that does make a difference in how we would recommend demoting one thing or promoting two species, not promoting, but changing two species.
So when you look at these records and decide just to change the name because somebody published it, what we find is when you actually go in and read the research, the conclusions are really
Right.
not very well done.
Yeah, and I'm actually doing that now. So like I enjoy pulling up Google Scholar whenever I can and reading the paper anyway that can get me back to the taxonomy. And there are times where I've talked with Mark, we don't think the dust has settled quite yet on this taxonomic uncertainty, so we just kind of leave it so we can save some labels. Oh, yes.
Have on the DNA sequencing for identifying species, has that been used to identify to cultivar level?
That's a good question, and that's really, I think, one thing Mark is interested is if we can have, you know, repeatable genetic markers that you can use across different taxa to get to that level where you're identifying cultivars. And the short answer is it's not really certain yet. So there was a paper published, I think, just a year ago from some botanic garden in Egypt where they're trying to use a combination of some of these barcoding genes to see if you can actually tease apart cultivars. And one thing I didn't really mention is I was using, say, 5 genes. Some of them are ribosomal genes that help encode proteins. They're just housekeeping genes. And then other ones are chloroplast housekeeping genes. They're helping, keep photosynthesis going. And the more variable you have genes, where you can tease apart species or even cultivars, the less likely it is to be conserved across a wide variety of taxa, which makes it less less valuable for people who kind of want to do exactly what you want to do across many plants in the garden. What we kind of have right now are, individual labs designing genetic markers that are tailored to their taxa of interest. So we have, very specific genes that can be used for this one group, and very specific genes that can be studied to study relationships of this group at the species level or even below the species level. Does that answer your question? I'm hoping that one day we will get to that point, and I want to be around when we do. So the the laborious part would be to maybe, instead of sample 3 barcode genes, sample 5 or 6 or 7, and the more sequence data you have, the more likely you are to have comparable things, to get more information to say if this is different from that. But then you get to this cost aspect, which is limiting. Good question.
Anybody else? Doesn't look like it. Thank you so
Thank
much,
you.
Andrew.